Assalamu alaikum. My name is Mohammed Hamza, roll number 63009 from MPhil Applied Chemistry, and my topic of presentation is types of nucleic acid. Here are the contents of my presentation. There are two types of nucleic acid, DNA and RNA. DNA stands for deoxyribonucleic acid, which containing the genetic instructions used in the development and functioning of all known living organisms. The DNA segments carrying this genetic information are called genes. DNA sequences are involved in regulating the use of this genetic information. The structure of DNA consists of two long polymers of simple units called nucleotides, with backbones made of sugars and phosphate groups joined by ester bonds. Attached to each sugar is one of four types of molecules called nucleobases, which is usually symbolized by a single letter A, T, G, C. As we can see that A stands the structure of DNA and RNA, adenine, guanine, cytosine, and thymine. It is the sequence of these four nucleobases along the backbone that encodes information. This information is read using the genetic code that specifies the sequence of amino acids with proteins. The code is read by copying stretches of DNA into the related nucleic acid RNA in a process called transcription. Here is the structure of pentose sugar, nitrogen base, and phosphate. The DNA double helix is stabilized primarily by two forces. The first one is hydrogen bonds between nucleotides and the base stacking interactions among aromatic nucleobases. These four bases are attached to the sugar phosphate to form a complete nucleotide. Adenine pairs with thymine and guanine pairs with cytosine. These two strands run in opposite directions to each other and therefore they are anti-parallel. Here in this diagram we can see that the sugar phosphate backbone nucleotide and the pentose sugar guanine is in anti-parallel direction to cytosine, and the hydrogen bonds are in dotted form. Schraff's rule states that DNA from any species of any organism should have a 1:1 protein base pair rule of purine and pyrimidine bases like adenine and thymine is equal to guanine and cytosine. More specifically, that the amount of guanine should be equal to the cytosine and the amount of adenine should be equal to thymine. The adenine and thymine are doubly bonded, having double bonds, and guanine and cytosine have triple bonds. Or we can say that triply bonded. In all species, it is composed of two helical chains bound to each other by hydrogen bonds. Both chains are coiled around the same axis and have the same pitch of 3.4 nanometer. We can see that in the diagram. The pair of chains have a radius of 2 nanometer. Here, two hydrogen bonds between adenine and thymine and 3 bonds of hydrogen between guanine and cytosine. Alternative structures of DNA. DNA exists in many possible conformations that include aDNA, bDNA, and Z forms. The conformation that DNA adopts depends on the hydration level, the DNA sequence, the amount and direction of supercoiling, chemical modifications of these bases, the type and concentration of metal ions, the presence of polyamines in solution. Here is the structure: aDNA, bDNA, and zDNA. We can say that a form of DNA. The aDNA is one of the possible double helical structures which DNA can adopt. The A form occurs under non-psychological conditions in partly dehydrated samples of DNA, while in the cell it may be produced in hybrid pairings of DNA and RNA strands and in enzyme DNA complexes, as we can see the diagram. The second one is B form of DNA. B form DNA is the right-handed double helix. The bDNA form is most common under the conditions found in cells. It is not a well-defined conformation, but a family of related DNA conformations that occur at the high hydration levels present in cells. The zDNA, or the Z-form DNA, is one of many possible double helical structures of DNA. It is left-handed double helical structure in which the helix winds to the left in a zigzagging pattern instead of the right, like the more common bDNA form. Segments of DNA where the bases have been chemically modified by methylation may undergo a large change in conformation and adopt the Z form. Technologies in which DNA used. The first one is genetic engineering. Modern biology and biochemistry make intense use of the techniques like phenol chloroform extraction and manipulation in laboratory, etc. These techniques are used to make recombinant DNA. Recombinant DNA is man-made DNA sequence that has been assembled from other DNA sequence. They can be transformed into organisms in the form of plasmids or appropriate format by using a viral vector. DNA profiling is the second technology. Forensic scientists can use DNA in blood, semen, skin, saliva, or hair found at a crime scene to identify a matching DNA of an individual, such as a perpetrator. This process is formally termed DNA profiling, also called DNA fingerprinting. In DNA profiling, the lengths of variable sections of repetitive DNA, such as short tandem repeats and mini satellites, are compared between people. This method is used an extremely reliable technique for identifying a matching DNA. Catalytic DNA, deoxyribose, also called catalytic DNA. DNA enzymes catalyze variety of chemical reactions, including RNA-DNA cleavage, RNA-DNA ligation, carbon-carbon bond formation. Bioinformatics. Bioinformatics involves the development of techniques to store data, mine, search, and manipulate biological data, including DNA nucleic acid sequence data. These have led to widely applied advances in computer science, especially string searching, algorithms, etc. DNA nanotechnology uses the unique molecular recognition properties of DNA and other nucleic acids to create self assembling branch DNA complexes with useful properties. DNA is thus used as a structural material rather than as a carrier of biological information. Information storage. DNA as a storage device for information has numerous potential since it has much higher storage density compared to electronic devices. However, high cost, slow read and write times, and insufficient reliability has prevented its practical use. The second type is RNA. RNA stands for ribonucleic acid. RNA functions in converting genetic information of genes into the amino acid sequences of proteins. As we can see that the replication of DNA transcription occurs and RNA forms. This RNA translation occurs and forms proteins. Here we have the RNA contains four bases: cytosine, guanine, adenine, and uracil, while DNA contains thymine. There are the three universal types of RNA include messenger RNA, ribosomal RNA, and transfer RNA. The messenger RNA acts to carry genetic sequence information between DNA and ribosomes, directing protein synthesis, and carries instructions from DNA in the nucleus to ribosome. Ribosomal RNA reads the DNA sequence and catalyzes peptide bond formation, and transfer RNA serves as the carrier molecule for amino acids to be used in protein synthesis and is responsible for decoding the mRNA. Codes in the form of amino acids for the synthesis of proteins for specific function called codons. Here we have three there. You can see that the image for peptide synthesis. In addition, there are other classes of RNA are now known. MicroRNA stands for miRNA. It is a small strand non-coding RNA molecule containing 22 nucleotides found in plants, animals, and some viruses that functions in RNA slicing and post transcriptional regulation of gene expression. The small interfering RNA. It is the class of RNA double-stranded non-coding RNA molecules, typically 20-27 base pairs in length similar to miRNA and operating within the RNA interference pathway. The other is RNA interference and double-stranded RNA. This term is used to describe a cellular mechanism that uses the gene's own DNA sequence of gene to turn it off, a process that researchers call slicing. A variety of organisms, including plants, animals, and fungi, RNAi is triggered by double-stranded RNA. The double-stranded RNA is a polyphyletic groups of viruses that have double-stranded genomes made of ribonucleic acid. The double-stranded genome is called to transcribed a positive strand RNA by the viral dependent RNA polymerase. Here are the applications of RNA like viral infection, neurological diseases, cancer, and difficulties in therapeutic applications. Antiviral treatment is one of the earliest proposed RNAi based medical applications, and two different types have been developed. The first type is to target viral RNAs. Many studies have shown that targeting viral RNAs can suppress the replication of numerous viruses, including HIV, hepatitis A, B, C, influenza, etc. And the cancer, a traditional chemotherapy can effectively kill cancer cells. Lack of specificity for discriminating normal cells and cancer cells in these treatments usually cause severe side effects. So, numerous studies have demonstrated that RNA interference can provide a more specific approach to inhibit tumor growth by targeting cancer related genes. Neurological diseases, RNA interference strategies also show potential for treating neuro neurodegenerative diseases. Studies in cells and in mouse have shown that specifically targeting amyloid beta producing genes by RNA interference can reduce the amount of a beta peptide, which is correlated with the cause of Alzheimer's disease. Difficulties in therapeutic applications. To achieve the clinical potential, RNA interference or siRNA must be efficiently transported to the cells of targeted tissues. However, there are various barriers that must be fixed before it can be used clinically. There is a question: how DNA directs protein synthesis? Double standard strand DNA in the cell nucleus. Here we have the first step. The strand of DNA unzip and allow free RNA nucleotides to link with the separated strands. In the second step, messenger RNA forming on DNA strands. Free RNA nucleotide approaches an unzipped DNA molecule to pair its base with DNA nucleotide. mRNA moves from the cell nucleus to ribosome in the cytoplasm. There it acts as a pattern on which amino acids transported by transfer RNA form protein. In the third step, the formation of protein, as we can see that the ribosome moves left to right along mRNA. The growing polypeptide chain eventually constitutes a protein, attached amino acid. This is how DNA directs protein synthesis. Nowadays, mRNA used to make vaccines for COVID-19. First of all, coronavirus like the one that causes COVID-19 are named for the crown like spikes on their surface, as we can see that in the picture, called spike proteins. These spike proteins are ideal targets for vaccine. The messenger RNA is genetic material that tells our body how to make proteins. The vaccine is made of mRNA wrapped in a coating that makes delivery easy and keeps the body from damaging it. The mRNA in the vaccine teaches our cells how to make copies of the spike protein. If you are exposed to the real virus later, your body will recognize it and know how it fight it off. The mRNA in the vaccine teaches our cells how to make copies of the spike protein. If we are exposed to the real virus later, our body will recognize it and know how to fight it off. Thank you. I hope you enjoy the whole assignment.